We went through about 50 different variants of the same molecule until we came up with the best one, and that was around 2012. Then we had our clinical candidate identified, and then we used the next 2 years for process optimization, for upstream process, choosing the right production cell lines, the transfection protocols, and so on, until we got a upstream process that was industry standard, the same that the big drug companies use for therapeutic amplification. And then a downstream process, purification, column chromatography, capture step, everything industry standard and scalable from small 5-liter reactors to 500 and 5,000 liters. And then after we had the purification process down and characterized the molecule by mass spectrometry, it was within 1 Dalton of the predicted molecular weight from the DNA sequence. So we were sure that it wasn't secondarily modified, no glycosylation and so on. We then spent a year and a half to identify an optimally suited formulation buffer. And after we had tested through about 50 or so different formulation buffers, we had one that worked very well, and which is also surprisingly only contains industry standard components, nothing exotic. And then we did a 2-year long-term stability study in the formulation buffer, and we showed that this agent is remarkably robust. You can keep it at -20, at +4, or at room temperature in formulation buffer on the shelf for 2 years, and it doesn't lose activity, it doesn't show aggregation, it doesn't show breakdown. It's like concrete. I say you can drive a tank over it and it still works. So we have a highly stable, unusually robust molecule, and we are trying to get with that into phase 1 clinical trials for AML patients. However, being a small startup company, we don't have the funds from where we are in late academic development into early clinical development. We would need something like 10-15 million euros. And investors have been approached. We have, in the last 4 or 5 years, we have had more than 100 investors discussions, but they were shying away because we don't have efficacy data in a mouse model yet, and we don't have toxicity data in a relevant species. So the conundrum is, how do you get somebody to finance something when you don't have efficacy data in an animal model? It's not that we have been lazy, but the animal models are more difficult for an agent like this, which recruits an effector cell rather than, let's say, an immunotoxin, which is toxic but doesn't rely on the presence of an effector cell. So if we wanted to have a xenotransplanted mouse model, they usually don't have NK cells, but our agent only works with NK cells, and our binding hits are uniquely specific for human NK cells and do not cross-react with the mouse NK cells. So you have to have a very elaborate model where you can xenotransplant a human leukemia into a mouse, and at that time it must not have NK cells because the NK cells reject it. And then later on you have to transplant the leukemia and then have to transplant the human effector cells, pre-activated, and then your agent. And that is such a complicated mouse model. They were not available until very recently. Now they are, and if we find something like 50,000-60,000 euros, the next thing we do is to do the efficacy studies in a mouse, but we don't have the 5 or 6 million euros that you need for a GMP batch from a contract manufacturing organization. So we are trying to get also some efficacy data and toxicity data with a cheaper protocol, and that is, in Germany we are in a unique situation. There is a law that permits compassionate use under specific conditions. A physician can treat an individual patient with an experimental drug. And we have some physicians that are willing to do that for us, although they must be very careful.